Hello everybody. So lipoproteins was actually a very confusing topic for me, and I didn't know where to study it from or like how I can go about it. But I found a video online on Ninja, and he basically summarized everything, and I just wanted to share my notes over what I've studied and gone through. So bear with me, it's going to be a little long, but I assure you that it's very useful. Okay, so the first lipoprotein that the body uses to transfer things from within the intestine, such as things that like cholesterol, triglycerides, or proteins, is something called the chylomicron. And what happens is that, let's say fats or triglycerides are in the small intestines, so they will be emulsified by the use of bile acids and bilirubin. And once they're emulsified, they will be broken down even more by protein lipase that is released from the pancreas. And then finally, after that, they will be packaged into bubbles or lipoproteins, and the first one here is chylomicron. And on this chylomicron, you will have a protein receptor molecule found here called the ApoB48. Once the chylomicron gets into the circulation or the blood, it probably meets with the HDL, and the HDL is a carrying lipoprotein. It takes care of all of the other lipoproteins and makes sure that the things like the LDL does not cause atherosclerosis, like I'll show you guys at the end of the video. So what it does, it shares its receptors so that it helps the chylomicron to bind to other places, to deliver the contents within itself to other places within the body, where they can use this product and use the energy, whether it is ATP, glucose, or whatever it is. So the two things that interact with other receptors that HDL give to chylomicron is C2 and E. Once it's given to them, the chylomicron, using C2 and E on its surface, can interact with a lipoprotein lipase, which is the receptor found on the tissues that first cuts the contents within the chylomicron triglyceride into glycerol and fatty acids. The sixth step here is that glycerol and fatty acids now will go into the adipose tissue or into the skeletal muscles to either be used as fatty acids or to convert it into triglycerides. Now for the seventh step, after chylomicron has interacted with lipoprotein lipase and gave its structures to those cells or tissues, it will give back its C2 as a seventh step towards the HDL. Now it will continue further and it will go to the liver. So after it gives its contents to adipose and skeletals, it will go to the liver as the eighth step. And so here in the liver, the E molecule on its surface will be able to interact with a different receptor called the LDL receptor and a LDL relative protein. And so here the chylomicron will be able to be taken into the liver. And so the 11th step here tells us what happens inside the liver. So break down the components. The cholesterol is used to produce bile salts, membrane use or storage. Proteins are broken into amino acids, triglycerides, glycerol, and fatty acids. Next, what happens is what's left inside the chylomicron will be packaged using B100 on the surface. And so when B100 is found on the surface, the chylomicron changes to a different lipoprotein called VLDL. So VLDL contains B100. So now again, HDL will come to it and give it the C2 and the E as well, so that it is able to interact with other parts of the body, just like how the chylomicron did it before. So now step number 15, VLDL, by the effect of C2 will interact with LPL, lipoprotein lipase. Remember, always C2 interacts with lipoprotein lipase. Then it will go, the triglycerides are broken by the lipase, as lipase is part of this receptor, and same thing, cholesterol and fatty acids are given off to the structures within the body that need it. So now the LDL becomes the remnant, and so it is also called as intermediate DL. Okay, so IDL now will contain on its surface the B100 and the E's, because already the C2 is used, that was used to interact with lipoprotein lipase. So IDL now will have the ability to either go back to the liver, or it will either go back to the go to a new place called the adrenal cortex. So in the adrenal cortex, the IDL will give its cholesterol for it to produce things like aldosterone and cortisol, and if it goes to the liver, it will also interact with something called the hepatic triglyceride lipase by its E receptor, and so same thing will happen. Triglycerides will be broken down into fatty acids and glycerol. Okay, and so step here 21, now the IDL having the B100 and E will have very low triglycerides, so it will give back its E to HDL, and so once it gives back its E to HDL, it becomes an LDL, and LDL now will have only a B100 on its surface. It can become very dangerous if this LDL accumulates. It's considered as bad, but one good thing, some good things about it is that it is used to interact with structures such as the gonads, such as the male and female gonads. In the male, it will allow for the production of testosterone because of its high cholesterol content, and the female for the production of estrogen and progesterone because of its high cholesterol functions. And the LDLs can also go to the liver by B100 interaction, and produce some stuff related to cholesterol, just like we have seen here. Also, LDL can also be taken by macrophages, where macrophages think that they are accumulating, so they go and in the place of inflammation, they go and eat in the LDLs. They keep eating those LDLs and accumulating them, and so macrophages turn into foam cells, and this results in atherosclerosis. So here, this is just a general idea of how LDLs function together and the whole process of it. I hope this was useful for all of you. Let me know if you guys have any questions, and I'll hopefully be able to answer them. Thank you very much. I'll see you guys in the next video.